So by now I think it's clear for everybody here that advances in neuroscience are intimately tied with advances in technology. There is not one without the other. And so we are asked the next speaker to expand even more on that. He's the managing director of the Neuroscience Center Zurich. His name is Dr. Wolfgang Knecht. Dr. Knecht, welcome to the stage. Welcome.
So, good morning, everybody. Today, I would like to give you a little bit of a glimpse of the Zurich perspective of neuroscience. And yesterday, we talked a lot about the networking, and that's what we do here. It's particularly my job to find synergies between the different disciplines that we have in our neuroscience center. Engineers, computer sciences, of course the clinicians take a very important part, and the basic neuroscientists. And the idea is that we have a lot of technology development and we have a lot of new knowledge within the last 20 years or so. When I was young, did my master's and my PhD about 20 years ago, we didn't have, for example, functional magnetic resonance imaging. This was just starting up. This is one of the most important tools that we have nowadays, and it's still in progress of development. We didn't really know a lot about brain plasticity. This is a very, very, very important potential that we have a lot of hopes right now in the clinic. When you go to the clinics, into the neuro rehabilitation clinics today, that's what doctors and professors want to understand. How does the brain actually reorganize when you have a brain injury? And these things have not been, I mean, we knew about it, but there's nothing been done in a systematic way to understand these mechanisms in more detail, because we didn't have the tools either. We didn't know about adult neural stem cells at that point. We heard that no, if you have an injury, no neurons can be produced in an adult brain. We learned in '98 that this paradigm is not true. This dogma is not true. So we have actually in very few sites of the brain and not too many newborn neurons in the adult brain, also in humans, which was shown in 1998. So there's a lot of development, and this is very exciting, but I still want to show you today that these things still take a very long time. And just to continue here, the problem that we're facing, you see here the numbers for Switzerland. They're incredibly high in neurology, particularly in psychiatry. We have the numbers for 2010 here. And the most important thing is that the causes and the mechanisms are mostly not clear. So that means we cannot really cure these diseases. We have therapies and they're advancing, but they're not really taking away the disease at the moment. So that stays the hard part of it. We talked also about the costs of brain disorders. We've seen these numbers yesterday. I want to add to this that brain research is also underfunded. When you compare the costs that are inburdened to society for diabetes and cancer and heart disease at the same time, add them up, this is about the costs that are being created and burdened to society by the diseases of the nervous system. So this is where we are in Zurich. You have here ETH, the main building, and just down the road you have the main building of University of Zurich. On the left side, you would have the University Hospital. It's the biggest hospital in Switzerland. And these are the three players that try to work together. And in Zurich, we had this very fortunate situation that all these three players are around for a long time. You see here the number of students. There's a couple of Nobel Prizes that we've gained. And also, very importantly, the number of spin-offs that ETH is creating is substantial. So this is the number since 1996. Most of these, actually 90% of those spin-off companies survive 5 or more years. These companies have created a lot of new positions and attracted a lot of venture capital over the years. We have the rankings. ETH is doing, of course, extremely well in its key competence engineering and technology, also very good in the life sciences. So these three players, they got together some time ago in 1998 and formed this joint competence center. It's a network of about 135 research groups located and scattered all over the area of Zurich. We have also not just the main campuses I showed you, we have also 2 research campuses of ETH and the university a little bit at the outside of Zurich. And on all these different locations we have neuroscientists. And the idea is to actually create these synergies amongst these groups. We have a very international PhD program with over 80% non-Swiss students coming from all over. It's a very lively and fantastic program that's helping us in our research. Very importantly here, as we talked yesterday, you have to have these very talented, very motivated, excellent single researchers that are the driving force and also the driving force in the networks. I think this is a key thing. We're very proud to have 8 of our researchers having ERC grants in the recent years. These I consider as small Nobel Prizes. For the advanced grant, a single researcher gets up to 2.4 million euros for his single research. Because of ETH, we have a lot of technology. Everything that can be built state-of-the-art is available and can be available very quickly. What we need can be supplied, and it's in the usual areas. So this is very important and very, very useful. To have an international perspective on this, we have now established collaborations with University of Oxford, with UCL London, and with McGill. And now I want to give you some examples of very interesting and I think also exceptional examples of research that we've done. Here's one that has a long, long timeline. You can see this here. It started very early in 1911 when Cajal stated very clearly, he's a Nobel Prize winner in 2006, I think, stated very clearly that if you have an injury of the central nervous system, there's no regrowth of, no regeneration of nervous fibers whatsoever. It took about 70 years, and in that time, this was the dogma, and there was nothing to do about it. We had no idea about the mechanisms. A dear friend, Alberto Aguayo from McGill, who we're collaborating with, discovered in the 80s a very interesting model that you have neurotrophic factors in the peripheral nervous system that would stimulate nervous fiber growth after an injury, but you don't find these neurotrophic factors in the CNS. It took another 10 years for Martin, a very dear colleague of mine, Martin Schwab, who discovered then in '89 a different mechanism of why there is no regeneration in the adult nervous system. He discovered a protein that he called NOGO, which inhibits the regeneration of an injured nerve fiber. Martin also developed then very early an antibody that would neutralize the effect of that NOGO inhibitor. So you see now a treatment, you see an untreated animal which has a spinal cord injury and hand function is impaired. It's a macaque monkey. You see the same experiment with a treated animal now after 58 days. So there's a food pellet in the drawer. They know that there's this food. And so the antibody treatment has been checked in the laboratories on many experiments over many years before a company then like Novartis was able or was willing to take this into the clinical phase. And they developed the antibodies, and they're doing now the whole clinical evaluation. And what happened here is, it's a very interesting story. In 2001, there was a first trial with an immunotherapy against Alzheimer's disease to remove Alzheimer's plaques, the beta amyloid, with an active immunization. And there were serious side effects, so the trials had to be stopped. And some of the patients developed really meningitis, and it was very serious. And the stop, that was really a very big disappointment for us then. Now, these patients were anyway very closely examined after the trials. And what people found out in Zurich is that there are antibodies. And that's the point. There are endogenous antibodies that were active against these amyloid plaques. They were found, but we had these side effects. So, and that then inspired actually a good colleague of mine, Roger Nitsch, who is leading all this research, to ask the question completely differently. It was not anymore, so what is the mechanism that these patients get sick? So what's happening in the diseased brain? It was different. The new question was, so what do healthy people do to not get sick? Because we have these elderly people, they're 70, 80, 90, and they're not getting sick. They don't have any dementia. So what is it that they have that other people that get sick don't have? So that was the question then they looked at. And that's the memory of the immune system, where you can see what kind of antibodies are produced during lifetime. So reading out the memory of the memory B cells then created, we found then actually new antibodies, endogenous antibodies that work against the Alzheimer's plaques. So back from the clinic to the labs, this kind of question was then for a few years really put back to the labs and were again examined with animal models that were treated then with an antibody coming from Roger Nitsch, created as an endogenous antibody, and it worked in the lab. So this would still take a couple of years, but also here, the timeline you see, the development, until you can translate or transfer results that you have into production, into pharma or biotech for production of the drug and validation of the drug, it takes really a long time. Are we? Okay, so a different field that we have built up since the last 8 years about is neurorehabilitation training, here mostly with stroke patients. The idea is that these training machines that we've created and partially already transferred to spin-off companies, the training is very important. You can intensify training of stroke patients, motor training. You can adaptively or adapt the performance to the development of the patient. Gaming, of course, is a very important aspect. These machines always have a gaming component. You play a game as a patient, and that will motivate you a lot to continue and to intensify this over weeks, which is sometimes necessary depending on what kind of patient or what kind of disease you have in particular. And here we come back to this plasticity that I said. The training and the parameters that we get out of these machines are then correlated with functional MRI to see the reorganization of the brain, to study this in a systematic way, which has not been done. And this is the effect that neurologists are, at least in Switzerland, are really concentrating on, because there's a huge potential if we understand how the plasticity, how these mechanisms work, and how we can influence them with the training. We will be able to optimize the training and to get most out of this of this kind of development that we've started. And we built this machine, which is also spin out now to a little company for assessment of these ataxia patients and for the disease progression. You see here, this is the trajectory mainly important for drug development in the more traditional way with immunotherapy, cell-based drug development, or molecules for certain drug targets. And then here, when it starts with the clinical trials, which are shown here in the different phases, then these things are really transferred to companies, and they do that. And at the interface here, there's the problem. And the problem is really at the moment that, and everybody knows that who works with interfaces, that there's a problem of communication, things get lost, things get misinterpreted, and things, many good ideas that we have here in academia are not making it to this part here anymore. So what we think at Zurich now is that we have to actually, from academia, develop further into this realm of industry. So in Zurich, we have a clinical trial center, and we have a very interesting first result here that has been recently in Science and Translational Medicine published in the summer. It's the first in-man phase 1 trial for this so-called therapy antigen-specific tolerance, I will tell you in a moment, in multiple sclerosis. So a very good colleague of mine, Roland, did GMP manufacturing of antigen-coupled cells. I will tell you in a moment. He administered them to only a few patients. He got all these regulations, permissions, and he did this trial showing that there's no adverse effects. It's just a phase 1 with a few patients. It's a proof of concept. So important here is the idea of antigen-specific tolerance, and that means that the immune system is specifically attacking, that we want to have the immune system being shut down when it specifically acts to the myelin and not to anything else. We don't want to shut down the whole immune system, but just the aggressivity to these peptides that we have in the myelin. And so Roland did that, and it was a very successful little example how we can push forward. We take the initiative now. If companies don't, then we do it. The idea here is what he did, that the antigen-specific tolerance would shut down the immune system just for those peptides that you find in the myelin. So what he did is he took out blood samples, he took out the white cells, the T cells, it's a sort of white blood cell, and he coupled them in the lab with 7 specific myelin-specific peptides and put them back into the patient. So once you have this coupling and you put these cells back into the patient, these cells undergo an apoptosis, so they die off after a couple of hours. And this signalizes in a certain way to the immune system to not attack those peptides that you find in the myelin. So the attack against the own myelin in the brain is reduced. That's the way it has been designed. And the success, we have very great hopes that we can really go into a next phase, and maybe also find partners who will join that. This is a last very interesting technology, only in a few seconds. It's magnetic, it's focused ultrasound that we use for surgery. This machine has been set up in Zurich, It is there for surgery, brain surgery. You can imagine that you have ultrasound around your head, 1,000 transducers. They will be focused, these sound waves will be focused in a small little region where you can do an ablation. Here it has been done for the first time in chronic pain patients. So this is technology that is very important for us. It's all experimental, but you can use that for all kinds of ablations and in the brain. And the obvious advantage is that there's no infection risk, which can be quite high. It's I heard that could be up to a couple % when you do, for example, stimulations, and you come in from the exterior. And we will start and design we have started to design a little project with W Investments to look at the data management and processing in the clinic, to really update the way we can present and then use the information also to bring it to the clinician. When he looks at these pictures, he gets much more readouts than just looking by inspection at these MRIs. We want to really introduce here the modern data management. As a last point, I want to summarize a little bit. So the goal is to really find novel and improved therapies for all kinds of brain diseases. We continue to strengthen the synergies that we have within Zurich and with our partners. It is very important that we think we need to create long-term relationships, and they have to be built on trust and on personal relationships, which is, I think, very important. We will continue to create structures to foster and promote this by having new training programs specifically designed for translational neuroscience, and we invite all students from the world, also from Saudi Arabia, to take part in this new PhD program. We need talented, motivated people. They're invited to Zurich. We will continue with these investigator-initiated trials. We will continue with our tandem professorships, and at the end, and this is a long-term process, regulations and politics plays a big role there, but at the moment the University Hospital in Zurich is being reorganized and enlarged, and within that process we hope that we can create a neuroscience campus which is directly next to the University Hospital, housing a lot of neuroscientists from the basic neurosciences, from ETH supplying us with technology. Thank you very much.
Thank you, Professor Knecht. Thank you.